When cells need more cells, they divide. In prokaryotes like bacteria, this is really simple. There is no nucleus, there are no membrane-bound organelles. They just duplicate the DNA and then divide in half. We call this binary fission, binary meaning two and fission meaning to break. In eukaryotes, things are a little more complicated. You have organelles you need to make copies of, and we need to make a copy of the nucleus. The nucleus is pretty complicated. It's a lot more complicated than the DNA in the prokaryotes. So we need a system, and we have one. It's called the cell cycle. The cell cycle is not just about division. The cell cycle is the whole life cycle of the cell, and the cell spends only a tiny little fraction of that whole cycle in cell division. Some cells, like the cells in the tips of growing plants or cells in embryos, are dividing very quickly, and so they have a very quick cell cycle. It's easier to see cell division in these cells. Cancer cells are like this. They divide very quickly. So one of the ways to disrupt cancer cells is to disrupt the cell cycle, and that's what chemotherapy tries to do. So the part of the cell cycle where the cell divides is called mitosis. We have divided mitosis up into stages, but those stages are kind of artificial. Mitosis is a process. You can think of it like the stages of life. It's easy to tell a toddler from a teenager from an adult. There's no distinct point at which a teenager becomes an adult. It's a gradual process. Mitosis is like that. It's a gradual process of moving from one stage to another. Sometimes you see a cell that's not obviously in one phase of mitosis or the other. It's possible for cells to be in in-between phases. Cells start off in interphase. That's what we call it when a cell is in between cell divisions. It looks like a normal cell that we've been looking at with a normal nucleus. That's a cell in interphase. Then the nucleus starts to go away. The stuff inside the nucleus, that grainy looking stuff, that's called chromatin. And it starts to condense. It starts to coil up with proteins, and it becomes what we call chromosomes. A chromosome is a piece of DNA that's all coiled up with proteins. And they are big enough that when you stain them properly, you can see them with a light microscope. We call this phase, the phase where the nucleus starts to disappear and the chromosomes start to appear, prophase. After prophase, the cells line up in the middle of the cell. Here you can see the chromosomes have two sides to them. Those two sides are identical, and so we call them sister chromatids. The sister chromatids are attached at a single point in the middle, and we call that the centromere. Now you can see that the centrioles have divided and moved to opposite ends of the cell. The centrioles start to form spindle fibers from the centriole to the centromere of each of the pairs of sister chromatids. When the chromosomes are all lined up in the middle like this, we call that metaphase. Those spindle fibers begin to shorten. They start to pull on the sister chromatids until they break apart. And one goes one way and the other one goes the other way. This phase, where the sister chromatids are moving away from each other in the cell, is called anaphase. Finally, the two sets of chromosomes have to reform into nuclei. So those chromosomes unwind and the nuclear membrane reforms. So you have the chromatin inside. And that is called telophase. Then we have to divide up the cytoplasm. The division of the cytoplasm is called cytokinesis. And now you have two new daughter cells, identical to the original first cell, but smaller. Remember, only eukaryotic cells undergo mitosis. And remember, mitosis is a continuous cycle. Those phases are not distinct. The cell moves directly from one into the next, and there's lots of in-between stuff going on. Stay awesome. I'll see you next time.